In this next video, we're going to cover how the brain controls our drive to breathe, as well as how that then is regulated during exercise. So, as you might imagine, we need relatively precise regulation in order to maintain homeostatic values for arterial oxygen content and acid-base balance, right? And so, in order to do that, we very, very tightly regulate both our inspiration and our expiration rate. Right. We know that from earlier in this unit that inspiration and expiration are produced by muscle contractions, right? Relaxation and contraction of the diaphragm. Diaphragm contracts, pulls out the ribs, expands the lungs. Air then comes in because of a difference in intrapleural pressure, and then as it relaxes, lungs compress again, intrapleural pressure greater than outside, and therefore expiration occurs, right? The specific part of the brain that's going to be controlling these motor neurons and accepting the afferent feedback from the system is located in the medulla oblongata. Yes, every time I still think of Waterboy jokes, every time I hear the medulla oblongata, right? And a small center of the pons, but medulla oblongata is really where we're going to focus. This is where our main respiratory center is located. It's interesting that we don't actually know all the mechanisms and fully understand how respiratory rate is controlled by this portion of the brain, mainly because doing these studies on humans is nearly impossible and unethical due to the sheer invasiveness of trying to measure how the medulla regulates this. So most of the information that we have comes from animal studies. So if we look at just a broad summary of the respiratory control, again, here's our motor cortex, which is going to control relaxation and contraction of the diaphragm and potentially of the accessory inspiratory and expiratory skeletal muscles. Right? So these are going to be controlled again by this respiratory center in the medulla. This respiratory center in the medulla is going to integrate feedback. So this is all afferent feedback here coming into the medulla, and then we'll generate a pattern that we can then use to talk to the motor cortex and tell us what type of contraction or the rate of contraction in which we need to develop in order to regulate breathing very closely. So here we use the word humoral receptors. What we mean by humoral is these are responsive to chemicals or substances in the blood. These are blood-borne substances is what this definition of humoral means, and there are receptors that are sensitive to these. We'll talk about them here more in a minute, but ultimately we have two different categories. So we have our central and our peripheral. So our central are actually going to be located in the brain stem and are going to respond to certain chemicals, and then our peripheral are going to exist all outside of this. The ones we're going to focus on here are going to be in the carotid bodies, but I also want to note that, as we talked about earlier, core body temperature will have an effect. So an increase in core body temperature will ultimately drive ventilation and increase that. As we saw, it will creep up in the last video. We have central chemoreceptors here working inside. There are chemoreceptors in the muscle that may be able to give us an idea of the metabolic properties and what's going on in the blood surrounding the muscle. The lung actually has stretch receptors and chemoreceptors, and then the carotid body and aortic arch. So let's focus there for now, because this is the perfect example of explaining humoral chemoreceptors. Again, this means found in the blood, right? And if we start to think about what potentially could act as chemicals that are going to activate these chemoreceptors, what you should be thinking is, of course, things are going to change with exercise, right? Located in the medulla, we can classify them as central chemoreceptors. So these are going to take place in the brain / spinal cord, right? This is central nervous system, CNS. Right. So our predominantly ones found in the respiratory control center of the medulla. It's going to be sensitive to the partial pressure of carbon dioxide and the increase in acidity or a drop in pH in the cerebral spinal fluid. When it sees this, a high PCO2 says that we need to breathe more to expire that, and we're therefore going to increase ventilation. In the peripheral body, what we'll see is aortic bodies are located in the aortic arch coming off the heart, and carotid located, as you might guess, in your carotid arteries. And again, if you think about why are these key sites chosen? Well, one, of course, the aorta makes sense from a physiological perspective because it's going to be indicative of arterial blood that's going to then go to the systemic nervous system. The carotid artery and the carotid body chemoreceptors are going to be more important and really going to serve as the key regulation for ventilatory control, because they control and monitor oxygen and PCO2, primarily PCO2 levels in the brain, right? And your brain is going to tightly control those so that we can keep it in homeostasis, right? So central nervous system responds to CO2 and increases in acid production or hydrogen ions, and will therefore, if any increase, we will increase the drive to breathe or increase ventilation. The aortic bodies and carotid bodies respond similarly to CO2, hydrogen ions, and then the carotid bodies also have the ability to somewhat respond to a change in oxygen. Again, having this near the brain, if we see a decrease in oxygen, then that would be important. And an increase in potassium, which is indicative of contractions, right? Potassium is released during, leaves the cell during an action potential and then can become a humoral chemoreceptor based on that. If any of these occur, increase in PCO2, decrease in O2, increase in acidity, or an increase in potassium levels, what we'll see is that there's an overall increase in ventilation in order to match this, right? These are indicative something of, in general, either something has gone wrong, or as far as this course is concerned, exercise is happening, right? We know increase in hydrogen and CO2 occur during exercise. So if we look at the effects of blood and partial pressure of CO2 and oxygen. Okay. So here we can see that as arterial PCO2 increases in millimeters of mercury, you'll note that normal is 40 millimeters of mercury in the arterial side, if we use some type of experimental procedure to increase the arterial PCO2 or partial pressure of CO2, that we will see a direct linear increase in ventilatory threshold. Right? What this indicates is that at rest, PCO2, partial pressure of CO2, is the primary determinant of your rate of breathing at rest. Right? And you can experiment with this, right? If you are so inclined, you can hold your breath, right? And as you hold your breath, if you feel like you want to open your mouth and breathe again, if you actually just blow out a little bit and expire CO2 and then close your mouth again without breathing anything in again, right, you'll be able to hold your breath longer, right? Or you could monitor it with some type of pulse ox, and you would see that your oxygen concentration, your PO2, very rarely changes, percent saturated hemoglobin, at rest. Right? So PCO2 is the important part. So in swimming and diving, this is any type of diving, there's a reason that people who are doing like free diving or anything will hyperventilate. They'll try to blow off as much CO2 as they can, and then therefore their drive to breathe will be suppressed. Right? Arterial PO2, if we alter the PO2 in the blood, our ventilation, you can see, doesn't really change. In general, we're going to be coming from this side, right? So arterial PO2 is normal, so in the alveoli, about 104, and hovers around 95 in the arterial side. Okay. So This is normal at rest for everyone is 95. As arterial PO2 drops, you'll see that we actually need a substantial drop, right? Small changes in arterial PCO2, large increase in ventilation. We need large drops in oxygen in order to see any type of change, right? And in general, this is really only significant at altitude, right? Where the partial pressure of oxygen is so low that you're actually having to breathe more in order to get enough liters of oxygen into your blood, right? And so we'll see that we remain relatively flat, and then about 65% or a PO2 of 65, then we'll see a rapid increase in the rate of breathing. But again, massive increases here in PO2 to drive any changes in ventilation. Tiny changes in PCO2 will drive rapid changes in ventilation, and that is an important take-home message. So if we look at the ventilatory control during exercise, we'll see that the primary drive for regulation of ventilation is due to the higher brain center, right? So that would, of course, be our medulla, our respiratory control center in the medulla. However, because we so tightly control our PCO2 levels, right, they remain relatively constant in homeostasis at this low intensity, submaximal, less than lactate threshold exercise, we know that there has to be some other kind of fine-tuning mechanisms to not over-adjust. These include our humoral chemoreceptors, as well as some neurofeedback from contracting skeletal muscle that will send signals in order to make sure that we perfectly match our ventilatory needs to our ventilatory supply. However, during heavy exercise, aka greater than lactate threshold, we see up to the point of heavy exercise, we see a linear rise in ventilatory threshold, and then remember we then see the rapid increase here in a nonlinear fashion. Okay? So what we believe that as we really expel the mechanism behind that is that there is an increase in blood lactate. Right? There's a reason that lactate threshold and ventilatory threshold occur very close to each other. Right? So because of this very tight coupling, it's believed that the major mechanism controlling the rate of ventilation during heavy exercise comes from the decrease in acidity in the blood, which then stimulates the carotid bodies to increase the rate of ventilation, or stimulates carotid bodies, which then talk back to the medulla, and the medulla then sends messages in order to increase the rate of ventilation. However, there are certain instances where there isn't this great association, and so it makes sense then that we have redundant factors in order to control that. So as we've mentioned in the figure before, those redundant factors can be potassium, body temperature, epinephrine and norepinephrine, may all contribute to this. So if we look at what happens with endurance training and reducing the ventilatory response. So again, we see a linear increase going from work rate in watts. Again, these are both relatively sub-max work rates. I know most students aren't familiar with what watts are, but most people's max is 250 to 300. So again, we're talking less than submaximal exercise here. So one thing that we can say is if we look at this curve, after training, you'll see that at given work rates, that after training, their ventilatory response, or the amount of liters per minute of air breathed, is less after training than before. Indeed, it's about 20% less than before. However, we do know that little to no effect of lung structure and function happens, right? The lung doesn't really adapt to exercise like most of the tissues that we talk about, which is kind of interesting. So then what's driving this? It's thought to be that it's due to a change in aerobic capacity of our muscles, right? So our muscles become more aerobic by increasing mitochondria. Therefore, they produce less hydrogen ions and lactic acid. we get less feedback from our muscle and our humoral chemoreceptors, and therefore we get less drive to our then motor signals being sent to our diaphragm are less, right? So again, normal lung is perfectly capable of absolutely exceeding any demand that we can put on it for gas exchange. We need no adaptation. However, it's interesting to note that that this isn't quite the case with everyone, right? In some elite, and I mean elite, high-end endurance athletes, as we talked about, training can result in hypoxemia. So why is that? So again, low to moderate intensity exercise, pulmonary system more than sufficient. We have no problems getting enough blood or enough oxygen in blood. At maximal exercise, it's historically been thought not to be a limitation in healthy individuals. And indeed, my advisor for my postdoc, Dr. Scott Powers, he worked on this for a long time, looking at ponies, actually looking at their respiratory system, because they have a very special, unique way of altering this, but looking at the regulatory control and if we could actually have a limitation. So new evidence actually suggests that there may be some respiratory muscle fatigue occurring at high intensity exercise. Again, not pulmonary system itself, not the lungs, but we may see some fatiguing of the muscles. Therefore, it may occur, or this may happen at about 90% really, really high intense exercise, right? And maybe in limiting endurance athletes. Indeed, about 40 to 50% of elite athletes, we're talking people who are winning marathons, who are Olympic athletes, may experience some type of hypoxemia due to this limitation on the pulmonary system. So to summarize up this section, the input to the respiratory control centers, mainly through humoral responses, are able to increase ventilation, can come from both neural, skeletal muscle, and humoral sources. Right? The neural input may come from higher brain centers, or it may arise from receptors in exercising muscles. Humoral input may arise from central receptors. These are in the CNS or peripheral, and or lung receptors. Our peripheral are going to primarily be our carotid and our aortic, with our carotid being the most important and really only the one sensitive to PO2. The central receptors are sensitive to PCO2 and pH, and the peripheral carotid are sensitive to PCO2 as well as the change in PO2, and are the only ones that are really receptive to that. And then to say this, the pulmonary system doesn't really limit exercise performance during prolonged submax exercise, but new evidence suggests that it may in elite athletes. So that'll finish it up for the respiratory system and exercise.